Hi, my name is Dr. Katja Klausz. I'm a senior scientist and I lead the myeloma research group at the Division of Antibody-Based Immunotherapy at the University Medical Center in Kiel, Germany.
Yes, and I'm Dorothee Winterberg. I'm also a scientist at the University Medical Center in Kiel, Schleswig-Holstein in Germany.
Okay, so maybe you know that you have an immune system and there are lots of different cell populations. For instance, T cells, NK cells, macrophages, and other subsets of immune cells. And normally they are activated against virus-infected cells or malignantly transformed cells. So if you develop cancer, your immune system is able, at least in the beginning, to eradicate these tumor cells. But at one time, this control is somehow lost. And now we only have medications that especially activate the T cell subsets. But we are working on other activating bispecific antibodies, so the principle is the same, but now we try to address the NK cells, which are potentially a little bit less harmful in terms of cytokine release syndrome or other neurotoxicities we see with the T cells. And yes, for more than 15 years, we are now working on the development of novel antibody-based immunotherapies for the treatment of cancer. During cancer progression, one big problem is that the tumor cells manage to evade the recognition by immune cells like T cells or NK cells. And therefore, we try to develop novel therapeutics to restore this recognition. And we design and characterize such antibodies and antibody-based immunotherapeutics that bring together, again, the immune cells to the tumor cells and, activate them to kill the tumor cells. And one of these projects is awarded with one of this year's Translational Research Awards from the International Myeloma Society and the Paula and Roger Riney Foundation. And here at the annual meeting, we have the honor to present our work. And this will be done by Dorothee, and she will now also explain to you a little bit more about the details.
Yes, thank you. Yes, I will give you a short summary of the project that we present here at the IMS meeting in Toronto. In this project, we designed, generated and characterized so-called NK cell engagers. These molecules can restore the immune recognition of multiple myeloma cells by NK cells. Naturally, NK cells get activated due to a set of activating and inhibitory receptors that sensing their specific ligands on stressed cells like tumor cells or also virus-infected cells. And on multiple myeloma cells, it is known that these activating ligands are usually upregulated. So the NK cells gets activated and subsequently kill the tumor cell. However, during the tumor progression, myeloma cells can lose these activating ligands, and these lead to that they evade the immune system. And this is where our NK cell engagers come to action, because they can recover this ligand receptor axis by simultaneously bind to a tumor-associated antigen, such as GPRC5D or CD38, and also to the activating NK cell receptor NKp30. And with this, they redirect the NK cells to the tumor site. And in our study, we see that our NK cell engagers induce high and very efficient tumor cell killing mediated by NK cells, but also induce phagocytosis by macrophages. And this is a frequent characteristic also described for other therapeutic antibodies such as daratumumab. We additionally see induction of cytokine release that was but however very much lower than this observed for the T-cell engager talquetamab, and this indicates that our NK cell engager still have an immune modulatory property that is important for the overall anti-tumor immune response, but without a high risk of the cytokine-induced cytotoxicity that is observed frequently for T-cell-based strategies. And although we have not clinically tested our NK cell engagers yet, we believe that NK cell engager can complement existing T-cell-based strategies and with this enhance potentially the overall treatment efficacy.